Zero versus missing versus blank. Don't mix them. Are you aware of the difference between zero, missing, and blank values in clinical trial study data? Let's explore. Zero. The real meaning numeric value indicating none, nothing, or zero quantity. It represents the actual measured or collected data. Say for example, I have number of adverse events which is equal to 0. That means 0 adverse events. Tumor size changes 0%. That means there is no change in the tumor size from change to baseline. Then the dose amount is equal to 0 mg, intentionally 0. So 0 is nothing but the actual data value. Missing. Data should exist but is not known, not collected, or not applicable. It is used in SAS and ADAM SDTM to represent missing numeric values. Say for example, I have the lab value which is not measured. So it represents missing. Subject withdrew before assessment. It represents missing. Equipment failure. So it represents missing. So missing is a value which was expected but we do not have it. Blank. Missing information in character variables. It is used for text fields where no value was recorded. Say for example, I have AE term missing which represents as blank. Reason for discontinuation not entered which represents as blank. So blank is generally the missing value for character variables. Summary. 0, it is generally used in numeric variables. It indicates the true value which is equal to 0. Say for example, dose is equal to 0 mg. Missing is used generally in numeric variables. It indicates data expected but not available. For example, say lab result is missing, so it represents dot. Blank is generally used for character variables. It indicates text not available. Reason is equal to blank. Most important points. 0 is not equal to missing. 0 is nothing but a meaningful data, whereas missing or blank is absence of data. Missing is only used for numeric variables, whereas blank is used for the character variables. In ADAM, always distinguish 0 values carefully because they influence analysis. Real-world scenarios. Real-world clinical trial examples that clearly show the difference between 0, missing, and blank values. Let's explore. Adverse events example. Scenario. A subject completed the study without having any adverse event. Say the value AE count is equal to 0. So it represents the subject truly had 0 adverse events. When AE term is equal to blank, that means no text was entered because no event occurred. 0 means no events happened. Blank is for a character field with no text. Concomitant medication example. Scenario. A subject was asked if they took any medications at baseline. Values. Number of medications is equal to 0. That means they truly took none of the concomitant medications. If the medication name is equal to blank, that means no medication name to record. If data was not collected due to site error, number is equal to missing. That represents we do not know is different from 0 medications. 0, it represents none, missing is equal to unknown, blank is equal to no text. Vital signs example. Scenario. Blood pressure reading was skipped due to patient fainting. Values say for example systolic BP is equal to missing. It was expected but was not collected. Heart rate is 0, rare but possible only if measured incorrectly, usually invalid. It represents like 0 would be a true value but usually clinically impossible. Missing because the data should exist but wasn't collected. Big mistake between 0 versus missing versus blank. Laboratory test example. Scenario. A sample was hemolyzed invalid. Lab values is equal to missing, which means lab could not provide a valid numeric result. Lab unit is blank. Units are empty because no result was generated. Missing for number and blank for character fields. Tumor response RECIST example. Scenario. Baseline tumor assessment was done. Say for example, values I have sum of diameters is equal to 0 mm, which indicates no measurable tumor present. If scan was not performed, that means sum is equal to missing. It was expected but was not available. If the response category is blank, that means no response category can be assigned if baseline scan is missing. 0 means tumor was absent, whereas missing means scan is missing. Study drug exposure example. Scenario. A subject missed a dose study. Values. Dose amount is equal to 0 mg. They were supposed to receive dose but intentionally did not take it. If the site forgot to record whether dose was taken, that means amount is equal to missing. Unknown if missed or taken. 0 is nothing but the actual 0 dose, whereas missing is nothing but unknown dose. Visit dates example. Scenario. Visit 3 date not recorded due to data entry error. Values. Visit date is missing for missing numeric SAS date expected but it is unknown. The valid visit description is blank. That means character field was left blank. Missing is for numeric unknown and blank is for character unknown. Real world scenarios SAGs example that demonstrate 0, missing and blank values exactly as they appear in clinical trial SDTM Adams trial data. Adverse events AE count versus text. Say I have a subject 1 with AE count is equal to 0. It represents subject truly had no adverse events. Now the second subject, the AE count is missing. That means AE data was not collected and it is unknown. Blank AE term, that means no text because no AE occurred or data missing. Laboratory results, numeric versus character. Say the first subject is having a valid lab result. The second subject lab value is missing. That means sample invalid or test was not performed. The third subject lab value is 0. That means true measured value, rare but valid. Exposure dose taken versus missing. The first subject took 50 mg. The second subject took 0 mg, that is missed dose intentionally, and the subject 3 is dose status is unknown. Tumor measurement RECIST scenario. The first subject is having 0 mm, which means no measurable tumor. Subject 2 is missing scan, that means cannot derive response. The third subject measurable tumor with response. Key takeaways for clinical programming. 0 affects statistics, missing excludes data from analysis, whereas blank is only for the character variables. Big mistake between 0 versus missing versus blank. Hence, ensure that you identify these terminologies, the difference of these values in the clinical trial data. I hope this video would help you. Many thanks for watching my video and happy learning. Please like, share, subscribe, leave a feedback comment. Many thanks for your support and having great connection.